Welcome back to another video, Mutation, Mutagenesis and Mutagens. This is Priya, Assistant Professor, Department of Biotechnology, SRMIST, Ramapuram. So coming on to mutation is a change that occurs in our DNA sequence either due to the mistakes when DNA is copied or as a result of environmental factors such as UV rays. So gene mutations have varying effects on Earth health depending on where they occur and whether they alter the function of an essential protein. So types of mutation, there are two main types of mutation, gene mutation and chromosomal mutation. So gene mutation is again subdivided into point mutation, frameshift mutation. So point mutation can be divided into three main categories: silent point mutation, missense point mutations, and nonsense point mutation. Whereas frameshift mutation can be subdivided into addition and deletion. So point mutation is a genetic mutation where a single nucleotide base can be changed or inserted or deleted from a sequence of DNA or RNA. Coming on to silent mutation, it's a mutation in DNA that do not have any observable effect on an organism's phenotype. They are a specific type of neutral mutation. So a nonsense mutation is also a change in one DNA base pair. Instead of substituting one amino acid for another, however, the altered DNA sequence prematurely signals that cell to stop building a protein. Missense mutation is a type of mutation in one DNA base pair that results in the substitution of one amino acids for another in a protein, thereby made by the gene. Frameshift mutation, this is another type of mutation occurs when the addition or deletion of DNA bases which may change the gene's reading frame. So frameshift mutation shifts from grouping of these bases and changes the code of amino acids. A deletion changes the number of DNA bases by removing a piece of DNA. Small insertion and insertion changes the number of DNA bases in a gene by adding a piece of DNA. Coming on to chromosome mutation, it's involved deletion, duplication, inversion, translocation. So deletion in the sense here, it's a type of mutation in which one or more nucleotides are lost or deleted from the chromosome. So duplication is an opposite to that of deletion, in which portion of genetic material or a chromosome can be duplicated or replicated resulting in the multiple copies of that particular region. Inversion mutation, if two breaks occur in one chromosome, sometimes the region between the breaks rotates 180 degrees before rejoining with two end fragments. Such an event creates a chromosomal mutation called as inversion. Translocation. So here the mutation takes place when the portion of G chromosome can be relocated. So the next topic is mutagenesis. It's just a process how the genetic information of an organism can be changed, resulting in the mutation. So mutagenesis can be various types: is directed mutagenesis, site-directed mutagenesis, PCR mutagenesis, insertional mutagenesis, signature tag mutagenesis, transposon mutagenesis, sequence saturation mutagenesis. So coming on to directed mutagenesis, it's also called as directed mutation, where an hypothesis proposing that organisms can respond to environmental stresses by orthogenetically directing mutation to certain genes or area of genome. So there are various types of directed mutagenesis like oligonucleotide directed mutagenesis with M13 DNA, oligonucleotide directed mutagenesis with plasmid DNA, and PCR amplified oligonucleotide directed mutagenesis. There are various advantages over directed mutagenesis, like its rate is high, all mutations can be induced, systematic and detailed, and its investigation of targeted mutation can be marked. Site-directed mutagenesis. Site-directed mutagenesis is a molecular biology method that is used to make a specific or intentional changes to the DNA sequence of a gene and any gene product. So it is also called as site-specific mutagenesis or oligonucleotide-directed mutagenesis. So next one is transposome mutagenesis. It is also called as transposition mutagenesis. It's a biological process that allows gene to be transferred to a host organism's chromosome, thereby interrupting or modifying the function of extant gene on a chromosome and thereby causing mutation. So this is a transposome mutagenesis where insertion of transposome within a gene leads to mutation. So the next topic is mutagens and the types of mutagens. So mutagens are nothing but the causative agents which are responsible to cause mutation in a normal gene or chromosome. So mutagens are normally known to be carcinogens which can cause cancer. So the mutagenic property of mutagens was first demonstrated in the year of 1927 by Muller when he discovered that X-rays can cause genetic mutation in fruit flies. So mutagens are divided into three main categories: physical, chemical, and biological. So physical mutagens are of two types: radiations and heat. Chemical mutagens are of five types: base analog, alkylating agents, intercalating agents, deaminating agents, and last one is metal ions. Whereas biological agents can be divided into three main categories: virus, bacteria, and transposome. So coming on to the mutagens types. First one is physical mutagens, that is radiation. So the first mutagenic agent was reported in the year of 1920. Radiation caused cross-linking of DNA or protein, chromosomal break, strand break, or loss of chromosome. At the molecular level, it can cause deletion of bases or DNA strand bases. Coming on to the X-ray radiation, it's one type of physical mutagen. X-rays are one of the most common type of ionizing radiation used in many of the medical practices. So it causes multiple strand break. If strand break occurs in both the strands, it becomes lethal for the particular cell. So UV rays, it's a non-ionizing type of radiation having less energy in it. Major causes of UV radiation are base deletion, strand breaking, breakage, cross-linking, and generation of nucleotide dimers. So coming on to the chemical mutagens, the first one is base analogs. So they are similar to that of bases of DNA purines. So there are two main base analogs which can cause chemical mutation in our body. So bromouracil and aminopurine. So bromouracil pairs with adenine as like that of thymine and causes mutation, whereas aminopurine cause adenine thymine to guanine cytosine or guanine cytosine to adenine thymine, thereby resulting in mutation during replication. So coming on to alkylating agents, so ethyl nitrosurea, mustard gas, and vinyl chloride are the main alkylating agents which can cause DNA damage. So these agents can cause DNA pairing errors by increasing ionizing and thereby producing gaps in DNA strand. Common alkylating agents are methyl hydrazine, temozolomide, busulfan, TEPA, dimethyl sulfate, ethyl ethane sulfate. So intercalating agents like ethylene bromide, proflavin, acridine, orange, or few intercalating agents, the molecules intercalate between bases of DNA and thereby disrupts its structure. Metal ions like nickel, chromium, cobalt, cadmium are common mutagens which can cause mutation. So coming on to the biological mutations, there are three main biological mutagens, virus, bacteria and transposon. So virus insert their DNA into our own genome and thereby disrupting the normal function of DNA or a gene. So once it inserts its DNA, the DNA is replicated, thereby transcribed and translated it viral protein instead of our own protein, for example HIV. Coming on to bacteria, some of the bacteria can cause inflammation. It prevents DNA damage and thereby it breaks the DNA. Transposomes, they are the non-coding DNA sequence which can jump from one place to another in a genome and influence the function of gene. Thank you all. We'll meet in another video.